Yes, YouTube is starting.
I should wait for them.
1 minute, 1 minute.
Yes, no problem, sir.
1 minute. In 30 seconds, I will start. Yes, thank you. We'll start now our lecture. Many again are joining. Yes. So let us start with the lecture given by Professor A.O. Ajay, who is a director, Center for Infectious Disease Control and Drug Development. He is in the Department of Microbiology, Adekunle Ajasin University. Now, it is difficult to pronounce me being an Indian. Akungwa, Akoka, Ondo State, Nigeria. So,
yes, sir.
Yes, he will be talking on use of selected medicinal plants and Chitosan bioactive component for infectious disease control. I don't know whether the organizer, ma'am, has joined or not. Hello.
Hello, sir. Before you continue, sir, the host should allow me to share because
yes,
if I want to share my slide, they have to correct that.
I will do it.
Okay, sir.
I will do it.
Okay, sir.
So now I have allowed you to present your PPT. Whether the organizer, ma'am, has joined? No. Okay, sir, you can start now sharing the PPT.
Okay, sir.
You can share your PPT. Screen
yes,
is available for you.
Okay, yeah. I'm trying on it now.
Yes. Yes, sir. Yes, sir. Yes, screen is visible.
Okay, yeah. Good everyone. I thank Professor Desmond. He's the president, Microbiologist Society, India, and he has been very active and versatile, and he has been playing a lot of encouraging role in the promotion of microbiology science globally. And that also informed the choice of meeting me as a guest lecturer on this particular occasion. I also thank Dr. Sujata Chaitanya, who is the organizing secretary. He has been linking up with me at least to make this presentation a reality. So this program was both, hosted and co-hosted by Swamin SSMW UTCO and Microbiology Society India. And the topic of my presentation is the use of medicinal plants and Chitosan bioactive components for infectious disease control. Well, we appreciate you, and by God's grace, and what informed our choice of this particular topic has to do with the current situation in our countries globally, and what we are experiencing in terms of epidemics and pandemic. And then gradually, well, we are just coming out of one, but well, we wouldn't wait till when we be having pandemics and even having different diseases ravaging our different communities, because on daily basis, many people are dying, and we try to at least overcome and make things more secured and make life more bearable through our scientific approach. The background of this particular lecture and research we have done so far has to do for the search of appropriate bioactive agents in overcoming the surge of infectious diseases globally, which is of importance and survival and for survival of a human race. And considering recent experience of coronavirus disease that ravaged the world, there is need to have some kind of dynamic approach in our health management system. I did a lot of work and investigation on various medicinal plants and related bioactive components for control of infectious disease and surveillance of antibiotic resistant reservoirs. This effort progresses from our university community at Adekunle Ajayi University, Ondo State, Nigeria, as well as during my postdoctoral research fellowship program at the University of Port Alice, South Africa. And this also cumulated to various discoveries and services rendered by me as the chairman post-COVID-19 free university community committee in my university here. And at least at the, we make the approach at the local level, and then also see what the effect it will have on the globally, so as to prevent the surge of different forms of diseases that is emanating on daily basis. Then, currently, as the director for Center for Infectious Disease Control and Drug Development of my institution, AAUA, I intensify effort to improve on existing investigation done by me to bring about formulation of more bioactive agents to make life more bearable for human race. During the control of, we thank God that the surge of COVID-19 is coming down, though we have some mutations that there are some resistant strains that are still coming up, but it's still better than before. And even whatever might be the case now, people should still be conscious anywhere they go to make sure that they protect themselves. It's not even the issue of COVID-19. There are so many viral diseases come up with serious infection than bacterial, protozoa, and some other forms. That's why people take much, much cognizance and efforts to make sure we safeguard people from getting infected with viral infections. When you talk of viral infections like Lassa fever, monkeypox, dengue fever, and so on and so forth, when you see the patients that are affected, you feel sorry for them, and it is very devastating because the mortality rate is also high compared with other forms of diseases that we can just use some common antibiotics to overcome. Okay. Then on the introduction, well, that just to let us know that on some review I made, because definitely when we are doing research, we have to make some review, some literature review to compare what we, to be able to plan and to compare the results of our research work with previous work done as well. So the potency of medicinal plants in eliminating etiologic agents depend on the abiotic components and the range of microorganisms they kill or inhibit. Thus, they can generally be classified as narrow spectrum or broad spectrum that antimicrobial agents that affect a wide range of microorganisms are broad spectrum. Anyway, that's just a basic microbiology or science that the case may be, because when some have specific group of organisms they attack, we just term them narrow spectrum, and there are some too that have a wide range. The wide range in the sense that it can be gram-positive or gram-negative bacteria or some other forms of diseases, whether viral or protozoa, as the case may be. So though we still have some specificity in that range too, just like the way we have some kind of broad range antifungal agents, as well as some other forms of antibiotics. And that's just has to do with the classifications, and we have many like that in microbiology. So it is also a vital note that some medicinal plants are valuable in boosting immune system so as to prevent the occurrence of diseases. In this realm too, when we talk of treatment is part of control, but even to prevent infections too is even more important, in the sense that and in preventing many infections, we have to boost our immune systems. So this has to do with some kind of antigenic situation or condition in our system. Then continue with the introduction. I just want to lay emphasis on global burden and solutions, and that's part of the focus and objective of this particular lecture and presentation. Antimicrobial agents are essentially important in reducing the global burden of infectious diseases. However, as resistant pathogens develop and spread, the effectiveness of antibiotics is diminished. This type of bacterial resistance in the antimicrobial agents poses a very serious threat to public health and for all kinds of antibiotics, including major last resort drugs. We mean that there are some commonly used drugs that are known to cure some ailments, but after some instances of use, maybe due to abuse or misuse, as the case may be, the target organisms will develop resistance to them. And that's one of the basic reasons too we are doing research day in, day out to find alternative therapies, especially as one of the aim of this study, which help us to determine the appropriate bioactive agents to enhance health safety and sustainable scientific development. And then when this is attained too, it also have so a good impact in our making in our economic recovery, as the case may be. So that has to do just making progress both scientifically and economically. In course of my investigations, there are various approach that we use. I put materials and methods for some reasons. Though the presentation, this lecture, encompasses is very wide, and as the case may be, I can also say that it touches so many areas that we have to use different method modified just to make sure that things works out well in making life bearable for people. So the study area for this research was carried out in Akumagba, Ondo State, though it's not limited to my university community in Nigeria, but there are some other areas too where I did some further work. But let me just give a brief description of this area, of this location. Ondo State is the western zone of Nigeria, western region, and the town itself is Akumba Akoko, and is within an Akoko land. There are some other small communities that was Then similarly, this particular university is formerly known as Ondo State University and was relocated to Akumba Akoko in 1999. So we have a reference for that, just to give you an insight of where we did this particular research and where my center is located. And I did some further studies too, during my postdoc program. That's at the University of Port Alice, that's South Africa. And this is near East London in the Eastern Cape region of South Africa. In South Africa, during my 1-year program, though I still went there for a few other things too, especially when my mentor there was doing his inaugural and so on. But in the course of my research, I was able to uncover so many things, at least in the surveillance of antibiotic resistant reservoirs, and come up with some genuine ideas on how to make life more bearable and find lasting solutions to some problems. Well, the next is the map of Nigeria showing the study site. So generally, the map of Nigeria, the map of Nigeria is as shown here, and then the area highlighted is the area highlighted in red is Ondo State, Nigeria, and that's where I'm currently talking from now. Then one issue on in most of our analysis, we normally collect samples, but samples in this realm now, when we talk of medicinal plants that we use for antimicrobial agent tests, there are different approach in collection for the sample. Then even the organisms we are testing them on, those are the ones that have the ability of causing diseases. We call them etiologic agents. So the medicinal plants used for these studies were collected from the wild as well as the research farm or Center for Infectious Disease Control and Drug Development, or the institution AAUA. So, well, when I was appointed the director of this center, there's a modification of my center. It was formerly called the Center for Biocomputing and Drug Development. But later on, we have to modify the name of the center because what we are doing is beyond the scope of just biocomputing. So, when we talk of molecular biology analysis, control of infectious disease, antimicrobial agents analysis, nanotechnology, water and food analysis. Then there's different forms of assays too, even soil testing and bioinformatics and other related works. So and then when we even look at the current trend of the global experience on pandemics, then we know that people need more help in certain areas just to make things easy and bearable. And that's informed our choice of changing the name of the center to Center for Infectious Disease Control and Drug Development. Then the microbial strains studied. In microbiology, basically, we have different approach for identification of microorganisms. Their conventional approach. Then we also have the PCR, that's the polymerase chain reaction, which is a molecular approach. And this gives us the precise idea for strain identification. That is, when you use PCR, you can be sure that we get 100% identification of the sample provided. Be it bacillus, enteric organisms, even from environmental sources, whether either other clinical or pharmaceutical sources for various purposes. So generally, the PCR helps us to give us precise identification. Then after that too, well, if a PCR at times too, maybe due to some challenges, like a power surge in our area, there are some of our samples too we collect clinically from different hospitals or health facilities, as the case may be. And well, when we have either a power challenge in storing our primers or some other facilities meant for molecular studies, Then we embark on the use of microbat, because microbat too can help us to identify up to 99%, close relatedness of the organism. So some can be 78, 79, even 80 or 90, which is still good enough. But when compared to the conventional ones, well, that one can, conventional, we call them probable identification. It's still basic too, but that's when we use badges manual, the case may be. Then similarly to API kits, because I remember in Nigeria, when we are doing that thing, when you use these ones too, they are a little bit costly, but not much. But with the current economy such too, all these things too, all these kits too are now even expensive. I can know the amount in naira, but I'm trying to correlate with what we have in dollars. So, but in essence, in basic microbiology, we use this approach even for my research to identify the microbial strains we are using for the study. The PCR, microbat, 24 e-kits, we also have 12 and so on and so forth. It depends on the rate, though I won't be talking much on this, but just to give a precise idea of what we did. Sorry, I think I was disconnected there. So, but I will still continue. There's a kind of power surge there. So I wouldn't know where the problem starts from, but let's continue to share my screen from where I started. Okay. Just a minute.
Yes, you take your own time. You can share.
Yes.
You are a co-host. You can share.
Okay. Yes. So I quickly want to go back to the last slide. The collection samples, microbial strain studied. Okay, yes, I was on the formulation of Chitosan. So on the Chitosan, well, we can get this from crayfish, or those, let me say crustacea generally. This crustacea can be prawn or crayfish, though that's where I started from. But even in the recent work I did in the extraction of Chitosan, I used a snail shell. But from previous work that I did and that was published, then when I even had some corpus with me and some even students on training, on serious training, I use this one to train them while the research is even going on, apart from some little workshop that we did thereafter. So in this regard, the raw materials in formulation of Chitosan, because when we talk of research, it's good for you to have some fundamental knowledge of what we did. On the Chitosan, well, in my investigation, I just noticed that suddenly find it valuable for various studies, especially when it relates on some of these antimicrobial agents and some other things. Though Chitosan is not a drug, but it can be used as a complement. So the raw materials that we work on, the case may be, that we use rather, is a prawn and crayfish, and this was processed by washing, drying, and homogenizing. And then the first stage, which I want you to take notes, maybe I should just say this to be clearer. There are three main stages for the synthesis of this Chitosan. So the three stages includes demineralization, deproteinization, and then deacetylation. So in the demineralization, I use 10% hydrochloric acid. Then we soak either the prawn and crayfish that separately into two samples for 24 hours at 60 degrees centigrade. Then this was washed and make sure the pH is balanced to neutral pH. After which I put the remaining samples in sodium hydroxide. That's 10% sodium hydroxide. And at that stage, it is called sodium hydroxide bleaching. That's a deproteinization. Then going further, that's the second stage, like I said. Going further is the use of sodium hydroxide with higher concentration. The second stage, which is deproteinization, I use 10% sodium hydroxide. And the last stage, which is the acetylation, we use 50% sodium hydroxide. So at this stage, that's the final stage where we wash, and the last residue we get is the chitosan. And at that point too, that thing used to degrade the bulk of the samples, the raw materials we use to be able to get the chitosan. And so like I said, I use both prawn and crayfish, but you can also use some other forms. Currently in my laboratory, I even have some commercially produced one. That ones we get them from shrimp. So I use that as a kind of can I say control, and some other research studies too, just to determine the effectiveness of what we manufacture compared with that one. Because by the time a bottle of about 25 milligram, let me even say a small bottle like that in Nigeria context is going to it's more than 25,000 naira or thereabout, but it can vary based on where you get them, and that will be around $50 or thereabout. So it's very expensive, and I feel that with some of these processes, if we have stable power supply, definitely something we can do. So, but at that last stage, which is the acetylation, it is just for 8 hours at 60 degrees centigrade, and but after that we wash with water and make sure the pH is stable at pH 7. Then we get our chitosan. Now, before we go to the analysis, then similarly in going further, we get our plant samples, then the chitosan. Then now we now go further on the systematic selection and control of etiologic agents. So in doing this, we try to determine the type of organisms we want to test them against. So like I earlier said later on, I said that we collected samples from various health facilities, and even in our surrounding area too. I remember at my center too, when you get to a stage, I said that we should, those that I'm training, that they should help me to get some samples from different areas, from the soil, maybe even from my family soil, from the hospital waste, and so on, and try to determine the nature of the organisms we have from those sources. And definitely, most of the organisms we test our antimicrobial agents on our clinical sources, and that's with the aim of determining whether this thing will be effective in case of infection by this type of organisms. So that's what leads us to the second stage of the systematic selection and control. So in this trend too, we did what is known as antibiotic susceptibility test. Microbiology, well, I think people that are experienced, even our undergraduates too at a lower level, many people are familiar with Bioactive antibiotic susceptibility test, but what we use is modified, and even beyond that. And nowadays, even when we want to quote, we go to the CDC and some other American bodies too, whereby they perform some standard tests recently. That's dated back to 2015-16, and I think we have some to even at 2018. So some of there are some modified approach to antibiotic susceptibility tests, either by agar disk or even through the use of broths, whereby we determine the effectiveness of the antimicrobial agents we are using. So on the sample preservation, well, definitely most of the samples you are using, there is no way you can say that you want to test something and you just finish your investigation or within a day or two. So we have to preserve them. So in most instances, most of these organisms too are kept in a refrigerator at 4 degrees centigrade just to slow down some metabolic activity. Anyway, these are basic scientific approach, even in microbiology, as well as some other related studies. But it's good that we take cognizance of it, and it's also important to mention briefly in this type of lecture. And in the context of determining our determining bioactive components, we also do what is known as phytochemical screening. So there are a lot of components that are being processed by plant sources, especially the medicinal ones. Though even those that are not medicinal, they are about, I don't know, there are different classes of plants, but we call them medicinal because they are valuable for, for treatment of one disease or the other. So some of those diseases may be of a microbiological origin, and some of them will be some other physiological causes. But even in some instances, when some of these components like flavonoids, tannins, flavotamins, and so on and so forth are present, they play one role or the other, either in eliminating some disease organisms or even improving some physiological conditions in the body that can aid the immune boosting system. So some of the phytochemical screening we did, which is also peculiar to some of these things, include flavonoids, tannins, flavotamins, alkanols, saponols. No, there are a host of them. So generally, I still feel that well, we do this one to make things more bearable and to determine the bioactive components of this particular plant sources. Then on the antimicrobial analysis. Okay, antimicrobial analysis, well, that was done using either the modified carbon method and some other related approach. Though I'm still going to talk much on this too, but we will also try to determine some other factors. Okay. Sorry, I'm still trying to. Okay. Now, medicinal plants. Well, this is one of the major things we are talking of. There are many medicinal plants with specific bioactive components valuable for target aimings, but some are commonly useful for resistant and recalcitrant microbes. Well, the only thing, I've been doing a lot of investigation on various medicinal plants and how to figure out some of them based on their activity. So including the ones figure out, because in the context of figuring them out may inform the activity or the role they have been playing at the local level. When people at the local level use them traditionally, and there are some of them too, well, maybe in some industries also try to see a way of using them, though there has not been success in some of them as using them as components of pharmaceutical industries. But when we come up with more ideas, we believe that we make more developments to make sure that some of these things are modified as pharmaceutical products that will help us to overcome some dangerous ailments. our populace. So among them is garlic, that's Allium sativum. And the bioactive components of garlic includes allicin, allicin, diallyl disulfide, and diallyl trisulfide. So there are few other components. So the issue of bioactive components is that, okay, let's talk of maybe allicin. There are some of them that can be active on this organism, and whereas some of the components can also be active on some other organism. That's the area too that we talk of bioactive components. That means it has to do with some compounds that are present in certain medicinal plants that makes it effective against certain diseases. So these compounds can vary. So those compounds you find in a plant, if you find them in some other plants too, that means those other plants too will be able to perform similar antimicrobial activity against certain organisms that we have been investigating. That's why at times too, when we find some kind of resistance surge, we try to go further and look for alternative therapy. We talk of chemotherapy. Some of our chemotherapeutic drugs, they come up with a lot of side effects. But going by some of these medicinal plants too, as per investigation, and even with my own observation and results we have in something, both in vivo and in vitro. Well, we see that because some of these things we are doing to, we also try some of them to in vitro. That is beyond the use of place in the laboratory, we test on animals and see the effects, their toxicity on some organs and so on and so forth. So that's why some of them comes up with less effect. But at the local level too, people should be careful because the erratic consumption of some, maybe some plants that we claim as medicinal plants too can also be dangerous. So we just have to be careful. And that's the main reason microbiologists and scientists all over the world are working around the clock just to find out, what are the pros and cons and the suitability of various antimicrobial agents. Then another plant that I just mentioned, before I go to that, on that garlic too, they use it in Africa, particularly in Nigeria, for various treatment of various diseases like abdominal discomfort, diarrhea, otitis media, that's ear infection, as well as respiratory tract infections. So amongst other ailments. Though definitely with my previous investigation, garlic too has been very much effective when I compared it with some other natural antimicrobial agents too. So, the only thing is the specificity. Well, it also has a kind of a wide range activity because some of the resistant strains we am using in my center, they are multi-drug resistant bacterial isolates. And then by the time we test something like this garlic on them, some of them are very susceptible. Without taking too much time on that, we also have Aframomum meliguata. In my local setting, they call it Atari. Well, I wouldn't be using some common names like that, but it's very common. No, I wouldn't say it's very common, but people really value it at traditional at the local level. Before I go to the benefits, let me quickly state some of the classes of compounds generally found in this Rafamamum. This include terpenoids, terpenoids, arachidonids, and flavonoids. Well, all these ones too, they are bioactive components that makes this particular plant effective. In those days, even when I was young, people, some people believe that they use it in protection against evil spirits. Even in some countries too, they have some other species, Aframomum. When we talk of some of these plant sources too, they come up with some species too. But the commonest we know is the Aframomum meliguata. And they said that they can protect them against some spirit when they chew. So anyway, we wouldn't say that one is scientific, but I'm just trying to go around the social link. Then the streets, the seeds, when crossed to, also have some as catalysts in some ethno-medicinal preparations against fever, skin diseases, abdominal pains, etc. We also have, after that, most of these plant sources I'm mentioning, their work I did some research on. So I will just, by the time I'm sharing the slides, I just show you briefly some of the areas I touched them. So because of our time, Andrographis paniculata, well, I wouldn't be mentioning some of the local names, but this one is generally known as the king of bitters. No, it's a bitter than bitter leaf, and truly, when I have tested it in my laboratory too against some of these resistant strains too, and find this to be effective against some of the strains. And some of the research too are in progress for publication. So I may not talk much on this too, but I just be giving you some of this peculiarity based on my observation and research. Then we also have what is known as Palanthus nunuri. Well, it may help to treat the urinary tract stones. There are some investigation done because I review some literatures too when I do research on some of these things, and which is normal. This one is, there are some investigation on it that they are dated to the year 2013. You know, that's it. But people are not even very much conversant with it. But we know that, the previous study just showed that it's effective against the herpes virus in cells. Though that Niruri, is less effective for that species. But when we do some kind of molecular docking, then we just observe that it is effective against gonorrhea and as well as COVID-19. That's why when I still go to an area where I just mentioned briefly the difference between wet laboratory and well, maybe we call the other one dry. And that's the area we talk about the molecular docking as the case may be. So other medicinal, okay. Other medicinal plants. Okay. Other selected medicinal plants, cannabis, sativa, protons and vesicles, Adazirata indica, Sonogratisima, Carita capaya, Jatruva feci. Though there are hosts of them. So, well, that's why I said my topic is wide and broad. And well, it's to give you the basic knowledge and insights of what is really going on at my center and the contribution we are making to microbiology society. Yes, and then to our society in general. And all these things we are doing to just to look, because you can't just say we work on one or two either plant sources and make a conclusion or derive an inference. So that's why we try on a host of medicinal plants that have been used locally and even scientifically and compare the activity against some etiologic agents. So like cannabis sativa too, I did an extensive research work with one of my research group in some other university in Nigeria, okay, including EXU, that's Ekiti State University, and few other universities like that. Though I have some collaborators, including Professor Adeyeye in EXU and so on and so forth like that. So we made some discovery and find out that cannabis sativa, apart from its use for some other physiological conditions, because I know that either women or men use it for some purposes, including growing of hair, apart from people abusing the drug and then they be stimulated, honestly. But the use for medicinal purposes at certain range can also be effective in curing some ailments. We have our data for that. So maybe at a later stage, I may still have opportunity of presenting specifically on some of them, because some of these things I'm mentioning are very broad and wide. But the basic things too, we just give people benefits of insight on the effectiveness of each of these medicinal plants. We have protons and vesicles. The cannabis sativa is what people is not everybody will go into science. It's the Indian hemp, so it has its own value, and has some antimicrobial activity. We have protons and vesicles too. Protons and vesicles, like in our local area, it's difficult to grow. Though we get some in the wild, and then some people too still manage to get in their area. At the local level, we by my dialect, we have a Yoruba name like Ajebo Kobale, which means that that Ajebo Kobale in English means witches dare not patch. That one too has to do with some local belief that some of these things also chase away some witchcraft and so on and so forth. So, but scientifically, what we are particular with has to do with the effectiveness on some disease-causing agents, which we also find very effective. And this particular plant, so there was a time I went to United States of America for conference, because I also belong to American Society of for Microbiology. So I presented it, and some of their technologies too. It's a PhD holder, and well, he specializes in trying to analyze various agents, including microbial agents and other things, which now he wants to prescribe some other tests that I can do on it, that I find my results very much valuable, and can really help to make some progress clinically. Well, that's why for some years now I've been working on that, and definitely we still continue on it, because one of the reasons why we even took it that is being used in combination of some plants at the local level to cure typhoid then. But at the end of the day, it is this particular protons and vesicles that we find valuable. That's why I continue the research on it. Then there are some other medicinal plants, Azadirachta, Indica, Ocimum glattissimum, Carica papaya, Diatrupa species. Well, they have their own bioactive components too. Then turmeric and ginger, they are rhizomatous because they are found underground. Turmeric is Cucumber longa, and ginger is Zingiber officinale. Well, they are very much valuable. I remember during this COVID-19 too, many people are taking a lot of ginger and so on, in the sense that what they believe it plays some valuable role in boosting the immune system and possibly alleviate some symptoms of COVID-19. So some of these things are very much paramount when we are doing investigation control of infectious diseases. I will not overrule the role played by some flowering plants like Cassepenia, Pulcherrima, that's a pride of Barbados, and Achillapa wickesiana, that's red Acalypha, commonly used for aesthetic purposes against some pathogens. That's why I'm just trying to move fast, considering our time, and make sure that I did some justice. And in the next slide too, I just give you the photo shot of some of these plants that I've mentioned. These are the samples of Aframoma meliguata, the seed, the leaf. Even the plant itself, this is how it looks. The seed, when you chew it, is really pepperish. It's pepperish. And then there are small seeds like that. So I've mentioned some of the values earlier. Then the Philanthus neruri, this is also the appearance. It appears like some of these plants too we find by the riverside. So actually, in my center, when I touch the leaf, it's very soft, and it doesn't take time to dry, because some of the plants we are using for studies, we usually dry them, blend them before we use different extraction solvents, to be able to extract different kind of antibioactive components or phytochemicals, as the case may be. And then these are the samples of the flowering plants I'm talking of. So, the pride of Barbados, that's Caesarpenia Pulcherrima, and so on and so forth. So, just to give you the appearance on how some of these things look. Then The other flowering plants, that's Acalipa ricketsiana. They have some other common names. That one can go by country anyway. The copper leaf and the postcode, as the case may be. And then similarly, the assays are performed on each of these medicinal plants. And in doing that too, we normally use some control. The control is are usually standard drugs. Some of these drugs are being used commonly for treatment of these diseases, or some of them are known to cure some of these diseases. And some of the antibiotics are used as control, include tetracycline and ofloxacin, which are emptied into 10 ml sterile distilled water to make up the talc solution of 225 milligram per ml. So at the end of the day, you get 0.2. We used to 0.2 ml of the standard drug, and that's therefore pipetted into the control well. So the in vitro demonstration of antimicrobial activity of the samples used, that the samples used, those are the medicinal plants and Chitosan. So the antimicrobial sensitivity test is done to know the ability of the antimicrobial agent to kill bacteria. Well, these are some of the publications that I made, antimicrobial properties of Aframanon meligataceae on Staphylococcus aureus and Streptococcus species. Well, I did this when I finished my postdoc degree program in SA. It's part of my pending work anyway, but because I take some interest in that particular work too. And that was dated to 2016. It was published in 2016. And that also informed the choice of the Dean of Science, then that he shows interest when he saw the publication that, okay, this particular plant will be very much valuable for antimicrobial therapy, at least for the means of disease causing organisms. Because that time we tested it on Staphylococcus and Streptococcus, which has are terrible pathogens, as the case may be. So we now try to look at the components and the other ways it can be used. And that leads us to some other work whereby you get funding to do extensive work. We form a research group to do that. And then even from our research group too, some also develop some other research from it. But in my university, I think in the faculty, in the science area, the biological test for this particular plants was started by me, and then I was encouraged to go further by my dean then. And we formed a natural research group that we try to improve on this. And then that leads from our research outcome, it leads us to publication of estimation of vitamins and Aframanon meligata and its relevance as immune booster. So that's why I said that at times too, when you see some of the components, some of them too can also aid to improve the immune system, whereby it just prevents the body being attacked or suppressed by the various forms of pathogens. Earlier too, I mentioned the issue of cannabis. These are some of, we also try to find, this is the research group I'm talking of at ESU. We did this all together, find various components of this cannabis. And we just find out that some of these components too can be valuable to eliminate some disease-causing organisms. Even after this publication, we made another one, but all these things will not be here, whereby I do the real general antimicrobial activity. But this one too, we just try to give us the components, and from what we derive from the components, it was published in Arctic Journal, which is also an high impact journal. And the result was very, we find it very interesting and valuable for us to proceed and do more studies on this. Similarly too, there are some other research that I did, that's antimicrobial activity of Zingiber officinale. That's a ginger on Staphylococcus aureus. Ginger on Staphylococcus aureus. So in this one, well, we can just say that the ginger, though I've mentioned it before, it has been found very valuable too in eliminating various disease-causing organisms. Staphylococcus and Streptococcus are pathogens. So at the initial stage of my investigation, I tried some of these disease-causing organisms of some known pathogens. Then these are published work or some research. There are extensive ones too, but that's why I said that I just pick some and be brief. Then going further on the qualitative method of analysis, most of our plants, generally for most of these studies, our filtrates were prepared by, it's not all of them that we boil. There are some of them we extract. So they are different. That's why I said that there are variation in the methodology. So the presentation, the lecture I'm giving is relatively complex that encompasses so many things. So that's why you can see some variation in the forms of approach. Like in some of them, I just use some extracts, but in few, like for this, like the one I did for, let me quickly, the one I did for, okay, I think it's, okay, it's for this ginger too, yes. Okay, the approach, we boil, there are some instances we boil the fresh plant for 5 minutes and filter through vacuum pump, and the filtrates are used for screening for flavonoids, tannins, and so on, alkaloids, reducing sugar, and anthraquinones, and anthocyanins. Then the test for alkaloids, that's about 2 mil, 0.2 mil. So anyway, some of these things too, I may not be reading because they are standard things. So, but we use few drops of brand Gendos reagents. We add it to it, and a change in color to orange to give us an orange precipitate indicate the presence of alkaloids. So tannins too, well, we have some reagents too. It means that in tannin, though in here I put ferric chloride means and the dark in color will change. But in some instances too, we may not even use the ferric chloride. That's why I said that there can be different approach. There are some instances we also use both sodium chloride and hydrochloric acid, following some sequence to get some of our results. So there are still some of the little variation. But at any rate, even if I'm publishing any of this journal, I just state the particular method or approach I use to determine the presence of these phytochemicals. Well, the other time too, we talk of the medicinal plants. It's not limited to the ones you get in the wild or even in our farmlands today, as case may be. Like some of the flowering plants too. The flowering plants like the Caesalpinia fucherrima. Okay. No, what I'm just trying to display for us is that some of these planet plants too are very much valuable in that they have some good activity against some disease-causing organisms. If you look at this chart, these particular flowering plants, I've already given the names and insights into some of them. I won't go back to that now. It has very much effective against Staphylococcus aureus. If you look at the bar, Staphylococcus aureus is the red, is indicated by the red bar. Then the lemon bar is Proteus vulgaris. So the 100% and 50% of this plant cells too is very effective if you compare with chloramphenicol, which is a known drug. So you can see that this particular medicinal plant too is which is also flowering plant. It's also almost competing with some known drug. And we are just considering in a situation that if you use this one, it will even have less side effects to compared with that chloramphenicol. Because even recently, there was a time even medical doctors used to prescribe chloramphenicol. But chloramphenicol, in recent years too, it has been discovered to be carcinogenic in nature. When you are treating a disease and you come up with cancer and some other terrible infection, you know that there's a problem. And those are the basis too. We find some of our research valuable, and then we see some other therapy that are very much effective, compared to the former antibiotics we are using. So in the study, the Tesapenia pucherrima, this is the flowering plants, exert some sort of inhibition. That's just have to do with the result I obtained locally. Well, when you talk of 20 mm, 16 mm, those are zones of inhibition, and then no majority of the people online here are microbiologists. And even as a layman too, all those ones just shows that the extracts we got from this plant source are effective against some disease-causing organisms, compared with the control value stated above. I've already explained that. And similarly to the second sample source. The in vitro antimicrobial activity of the medicinal plants, so such as Pramomum, Andrographis, and others as listed, and some nanoparticles that emanate from these sources is vital for the purpose. On the issue of nanoparticles, I will not, I didn't embed it with this particular lecture, because I did a research too that they are very much effective. It's part of my ongoing research. And I have reasons not even speaking on it now, because it will make this work too extensive for me. Basically, the use of nanoparticles too, though we have what is even known as a nanotechnology, which is one of the happening areas or developing areas of microbiology and biotechnology or science in general, because even most of the drugs that antibiotics, they cannot, most of the disease they cannot treat. The use of some nanoparticles is able to overcome some of those problems. Even the nanoparticles too also comes off because there is nothing that have benefit to that may not have some little side effect or problem. It has its own disadvantage too. Well, I think that has to do with the area of carcinogen or so on. But I feel that we can still do some study to maybe some of those problems can be neutralized. So that's why I said that I can't talk much on that. But nanoparticles too have been very much useful in some of our research studies. Similarly, the systemic selection and control of etiologic agents, which conceptualizes on the antimicrobial susceptibility test and sample evaluation, was considered. And we have spoken on that before. Then complementary to this, the source and dilution or standard drugs such as tetracycline, well, I think as consider that too. So I wouldn't like to take us our time. They are basic things. On the issue of the clinical isolate use, then you said they are varied and diverse. When I was in South Africa, we got some animal sources. We also have what is known as zoonotic diseases. That's why one should be careful the transmission of disease from animal to man too. And we just discovered that some of the organisms too that is being transferred to cause infection can be very grave. And when I'm back to Nigeria too, we did some further investigation. And some of the observation and the research we are trying to do in this regard is to just determine the resistance, what informs the possibility or what are the reasons why this particular disease-causing organisms are able to resist some antibiotics. So under this one now, when we are doing some kind of molecular analysis, we're trying to determine the composition of some resistant gene. So in this one now, the plate 5 shows the gel. The gel showing MEC-A gene at amplicon size of 310. Though there can be variation, which is big, and it's showing why some of these disease. Though we usually do some of these things for strains that are resistant, that are multi-drug resistant. And at the same time too, okay. So with that, we'll be able to determine what can be done. So we in that particular place I showed you to, when we analyze it, there is presence of the MEC-A gene, which shows the resistance to methicillin, to the use of methicillin drugs. No, we have what is known as a methicillin-resistant Staphylococcus aureus and so on and so forth. But by the time you characterize for the presence of vancomycin-resistant gene, vancomycin A and B, so they are found to be present. So in the medical clinical realm too, well, some of these things too will determine whether we are on course. And we also inform our choice of using certain antimicrobial agents, whether the antimicrobial agents we are using is appropriate or not. And if I can go back to the history, there was a time too when we started with the use of medicinal plants and even antibiotics generally. When people asking diseases, they recommend methicillin. But when there's resistance to methicillin, then medical doctor can say, okay, go for vancomycin. Later on, antibiotics, some of these organisms too, especially the skin microflora causing skin infection, get resistance to vancomycin. Then they change drugs. Then we have the class of fluoroquinolones. Well, people are happy that, okay, well, another class is coming, nothing can overcome it. Then before we know it, there's problem. That's why we now fluoroquinolones class 1, 2, 3, and so on. So at the end of the day, the class 1 is not functional 2, then 3. So we have the class like cephalosporin and so on and so forth that was effective. So what I'm just trying to say is that there's a kind of a chronology of a resistance. With that, we are changing. We can just continue to be changing some of these drugs to find the ones that are effective. And that also makes the research and work on medicinal plants and some other bioactive components, even from Chitosan or some other sources, to be very much valuable and overcome some of these disease and such. Then most of the things I've been talking on so far on wet laboratory, while I'm time conscious, I quickly just go through some few other areas too. And then that will not take much time again. And I just talk straight. So the wet laboratory just has to do with what we are actually seeing physically on using our plates and maybe lab animals, both in vivo and in vitro and so on and so forth. But talking about dry lab, on the other way around too, we do what is molecular docking, whereby some of these things are programmed on the computer system just to determine the bioactive components. And then on our Chitosan bioactivity, well, it's a polymer that is suitable for various biological, clinical, and industrial applications based on its excellent properties such as biocompatibility, biodegradability, and then it also has some homeostatic. It also has some homeostatic and hydrating properties, whereby it's useful for wound management, including anti-inflammatory properties. And Chitosan nanoparticles too are used as a drug carrier. I've mentioned something on the possibility of using some nanoparticles. And recently, I think about is it 2 weeks ago? Okay, it's going to about a month now. I coupled some nanoparticles with Chitosan, and with that, I find them very much effective against most of these multi-drug resistance clinical isolates I'm working with in my center. That's why I said that there are a lot on that, so I won't speak on that now in this presentation. Then chitin and its the acetylated derivative, that's chitosan, varies in linked acetyl amino and glucose residues. Well, in most instances, most of the animal sources, that is the cosacea we use for as raw materials for chitosan, they possess chitin. And I said I also tried some alternative using snail shell. That one is even relatively difficult because the snail shell is so stubborn and even difficult to degrade by some of this chemical approach. Though apart from the flow chart I showed you earlier, there are some instances too. I learned there's another approach whereby you can boil those shells and so on and so forth. Well, but in essence, what we just have to know is that it has a very good, it works in very good synergy even with some of these plant sources. Then molecular docking for bioactive components. The molecular docking was intensified by doing some works using Chimera software. The protein that's downloaded from a protein database, that's PDP, was imported on Chimera. This was followed by selection of chain. Well, this can be A and B, but chain A was selected. No, some of the things I just put in the slide here just has to show the sequence of molecular docking. And then the essence, the advantage of molecular docking is that it helps us to know the affinity of the ligands we download from our antimicrobial agents. So I need to summarize this because of our time. The ligands that we now dock against the protein, and then they have some affinity. So at times the lower the affinity, the better. And some of these things also gave us the inference of the possibility of using certain anti-medicinal plants against certain disease-causing organisms. Well, this is a typical table. This is okay, and the molecular docking, we use some package like Pyrex, like Pyrex, Discovery Studio, and so on and so forth. So I said I'm trying to move faster, considering that I think we've used over an hour. Then for this, okay, this is a typical table or the docking score of the molecular docking. That's Simum gratissimum against Escherichia coli. It's part of my ongoing project or research work. And I take much interest on the possibility of the immune boosting property of some of these plant sources, as well as their antimicrobial properties. Like I said, I've spoken on some of these things too, and under the West Laboratory, the molecular docking done on Philanthus nenuri, plants to extract as antimicrobial agents against that's the protein of Neisseria gonorrhea. So, well, we found that I tag the name of the ligand there, which is the terpenes as an active phytochemical component in Philanthus nenuri, as the highest binding affinity to purine in Neisseria gonorrhea. That purine is a protein in the organism. So that's why I said that we usually dock the ligand from the active from certain plants against the protein of the organism we are testing. So that's why from our molecular docking now, it shows that there's higher probability that Philanthus nenuri plant extract can be used as antimicrobial agent against Neisseria gonorrhea that causes gonorrhea diseases. Then similarly to, well, during pandemic, there was a significant use of medicinal plants for both prevention and treatment. Respondents and treatment. Respondents participated in the study, reported that they used medicinal plants as preventives and treatments related symptoms. So no, there are diversified suggestions by people, but well, at our own hand here as scientists, we are also doing our investigation to make sure that when we don't abuse the use of some of these things. The protein for SARS coronavirus spike protein that 2AJ5. So when we talk of some of these organisms, they usually have a kind of protein or receptor site, and that will determine whether an antibiotic or antimicrobial use against them will work or not. So the ligands for protons by tambecicus includes camphor, osimum, gratissimum. One of the bioactive components includes eugenol, Syngiba, officinal, terpene, then cassia, fistula, that's camphenol, etc. So there are also them like that. Then generally, we talk of bioactivity. So well, I wouldn't say I replace this one with discussion. No, it's a context for me to round up my presentation. The antimicrobial analysis of some extracts showed bactericidal and bacteriostatic effects, which is also inhibitory antimicrobial effect as reported by the Gonedinmale et al. That's 2018, thereby confirming the efficacy levels of studied medicinal plants, which is 2019. So in this context, I'm just trying to relate what I did compared with the past work done in literatures. So plants might have bacteriostatic effects. It might still be effective against microorganisms. Thereby, other factors such as exploring different solvents and other microorganisms before conclusion is important. So what I mean by this is that there are some instances, some of these plant sources, we look at them as not being much effective. But when we use different solvents, for example, in extraction, we use different solvents like ethanol, methanol, petroleum ether, N-hexane, chloroform, and so on. So most of these solvents extract different things. Even if you use water, you can also extract different things. And then the different bioactive component they extract will determine the activity on the test organism we are using. So, and then when we talk of bacteriostatic, inhibiting growth, or bactericidal, that's killing the organism entirely. So there are little differences. Well, I didn't need to be spending this one too much, but at the same time just for a better understanding. So some of these things, the use of different solvents to determine the effectiveness of some of these plant sources when we use solvents and then try them on different clinical isolates. So in conclusion, to round up this presentation, I come up briefly with some observations, which will not take time again. This research work supported the traditional use of many medicinal plants itemized in this study for treatment of appropriate ailments. The complementary use of Chitosan, which is a biopolymer, is also found valuable for its ability to reduce infectivity when synergized with some potent medicinal plant. So, like most of the research I did here, at the end of the day, I work on what is known as synergy, when I maybe try to medicinal plants together and see the effectiveness on plants, or even try them with the Chitosan. So even there are some of them when I put little amount of Chitosan, it shows a very good activity compared with when they are being used singularly. That's what we call synergy. It still shows that the use of Chitosan is very much relevant even in the same certain realm. Well, I'm still doing further investigation on this particular biopolymer because for some other modifications, which I still believe to able to serve as kind of a potential components for some pharmaceutical products. However, pharmacological standardization and clinical evaluation of medicinal plants are essential to make the natural products discussed in this context to be standard remedies that could effectively combat some mutated pathogens that have developed resistance against antibiotics abuse. Study of Chitosan has proven its use to be valuable for clinical applications. So, well, I've spoken, on that, and I still believe it has a very much valuable use. So, the surge of multi-drug antibiotic resistance necessitate the need for bioactive components from natural sources. Well, Chitosan is not from plant source. It's another, like I said, it's a biopolymer, and that is used, compared with other medicinal plants. And most of these medicinal plants too, where you can just name them, but they have certain components in them that makes them work against certain diseases, or maybe if it is a wide range of diseases, when we call them burst return. So those components that makes them effective, or enhance their potency, their bioactive components, which are what we have extensively discussed in this realm. Screening plant extracts for antimicrobial activity shows that higher plants represent a promising source of new antimicrobial agents. Plants produce a number of substances, and most are secondary metabolites. So medicines derived from plants have no apparent side effects that are associated with modern drugs. So, some of these things too, in conclusion, are justifying, the potentiation of this research outcome. And then generally, too, medicinal agents, in particular, antimicrobial properties of plants are attributed to the presence of active components, such as polyphenols, including flavonoids, alkaloids, and other compounds. And formerly, when I even try to describe some components peculiar to various plants, such as the eugenol, that makes grattimol active, or the camphor, or the terpene in Philanthus and urea, and so on. Those ones are bioactive components of medicinal plants, and they have been very much found valuable for various to eliminate various disease causing organisms. So in this realm too, our research on the use of these selective medicinal plants and Chitosan bioactive components for infectious disease control is justified, and will help us to keep human race safe from various disease ravaging around the world. Thank you very much. These are my references, and thank you for listening. Thanks.
Thank you, sir.
Yes.
Thank you, sir.
Yes.
First of all, on behalf of Microbiology Society India, I thank Professor Evo Ajayi, Director, Center for Infectious Diseases Control and Drug Development. Really, that was a very great talk. It's a really honor for you to share your valuable research. So now the session is open for discussion. If you have any discussion, please ask Professor Evo Ajayi. And it's really great to learn certain developments, because after the pandemic situation, everyone is looking for alternative antimicrobials. Really, it's a great work. If you have any discussion, any questions, please.
Yes, sir, you have delivered a very nice and elaborative lecture.
Thank you, sir.
It was in detail.
Yes.
And this now it is recorded in the YouTube. And some of the students from India, for example, Appa Patil, he is working on the same line. So he joined to
Yes, sir.
he attended your lecture.
Okay, sir.
So again, I will reiterate that in Nigeria, we want to continue the serving microbiology subject.
Okay.
So I will request again, you take a lead and we'll work together with the other teachers from Nigeria
Okay.
and we'll serve the student. Our main purpose is to serve the students.
Okay.
Now, among the attendee, anyone want to ask the question? They can. Otherwise, ma'am, you can conclude. Sujita, ma'am, Dr. Sujita.
Yeah, as our president Deshmukh sir told, it's really great to work on herbal antimicrobials. It's really because
Thank you, sir.
yeah, biopolymers are
Yes. Someone
they-
want to ask the question.
yeah.
Yes, you can ask, please. I think Ayotunde want to ask the question.
No, I'm not interested in.
Yes, you can.
I'm not asking any questions, sir. Thanks.
Okay. Then you continue, Sujita.
Yeah, so in future, definitely Microbiology Society of India, it will work with the Nigerian professors and team to come up with a solution for a worldwide problem, that is antimicrobial resistance. Yeah, someone
Okay.
wants to ask. Think.
Would be awesome.
Yeah, if you.
Okay, Yawan. Okay, Yawan. You can omit your speaker.
Thank you, Prof, for your. Thank you for the elaborate. Hello, can you hear me?
Yes, I can hear you.
Thank you for the elaborate presentation and for the expository information on medicinal plants and uses and some of
Okay.
your exploits. We
Yeah.
appreciate the lecture. So my question is concerning the topic of this presentation. I was hoping that I would look at some of the exploits you have been able to garner via this Chitosan. Maybe you have been able to carry out some investigation, be able to give us the exact point at which Chitosan is actually, playing the role it's playing. Maybe as an adjuvant, since we know that it's not actually an antimicrobial agent, but I think it plays a role in enhancing antimicrobial activity of certain plants. Maybe you have done something and you have been able to share with us. That was my question. I was looking forward to see. Maybe you can tell us regarding this. Thank you.
Okay. Thanks a lot. On the context of the Chitosan, there have been a lot of work that have done on it. But to be precise, I said that the topic I'm treating is very complex and elaborate. And, even within the scope of what I did, we've already spent over, almost close to 2 hours now. But on the Chitosan, I worked on it, and it works on, generally, it has low microbial activity on microorganisms. But when used in synergy with some medicinal plants, though I mentioned some of this thing briefly, when used in synergy, the synergy means that possibly maybe we use about, let's say 10% of Chitosan with the sample of antimicrobial extract. So some of those synergized samples, when we try them on some of these clinical isolates, we find them to be more effective. So even at my center, when I have some purpose with them, we work on that realm, and it was also published. That's why I said that. I have another what do you call it? Another package for even some research work in progress. So that's why I didn't talk much on the synergy of Chitosan. But it's very much effective. It has some antimicrobial property. Even I'm even looking to some ways that you can use it for certain packaging, but it's still an ongoing project. That's why I didn't talk too much on them. So, but specifically in my study, I tested that Chitosan both from plant sources, synthesized from plant sources and from crayfish separately. And then we test them on some of those clinical isolates. Then after that, I also synergize them with some of the medicinal plants. But ordinarily, the one from plant sources, when I test them, I think it shows more activity in most cases. But by the time I now synergize them, maybe by using 1%, because I remember the last research work we did on it, 1% of the Chitosan, synergize with some medicinal plant source. Medicinal plant source such as, let me quickly double check. There are many of them because I have we have a medicinal plant source such as the Osimum gratissimum and even Aframomum meliguata, and there are hosts of them like that. But in essence, in short, the discovery we have is that the synergized components is very much effective than using them alone. That's why I said that the Chitosan itself is not a drug, but it can be used as a complement. And when I even went for that on that, I said that there is even an ongoing research, because as at last month, I used some nanoparticles with it, and that one shows an excellent result. But well, that one that I did, the result is exceedingly it makes me, I felt delighted with it. But that thing too, there may be need for some other further tests, which may also be in vivo because of trying to look at what could be the adverse effect. So that's why I said that when you are talking of the specific result on that, well, I try to state some things here, but I did not go further on that. If I go in displaying some of the results we have, then it will take us double of the time. But in essence, particularly on the Chitosan, even if I'm to deliver a lecture on the antimicrobial activity on Chitosan alone, it will even be enough as much as what I did today. So that's why I generalize everything. But in essence, talking of specificity, the Chitosan too have been very much effective. And I also stated the processes that we undergo with the extraction. That has to do with the stages of demineralization, deproteinization, and then the acetylation. And in the demineralization, I said that we use 10% of sodium hydroxide, and then at 60 degrees centigrade for 24 hours. In the deproteinization, that's okay. The first one is 10% sodium hydrochloric acid. I mean hydrochloric acid. The deproteinization is a 10% sodium hydroxide, and the acetylation, which is just for 8 hours at 60 degrees centigrade, is a 50% sodium hydroxide. At this time, most of the samples that we use, whether it is crayfish or prawn, will be really degraded, and what will be left with is Chitosan. So even at the initial stage when we did the work, we have almost a bucket of crayfish, and what we got there is just a little quantity of the Chitosan at the end of the experiment. And this is what we use to test. And specifically, like I told you, the antimicrobial effect is low on its own. But when coupled or used in synergy with either plant sources or nanoparticles specifically, well, you get an exceedingly very fine results that we can really quantify and then work further on. So specifically, I can state is is a very good area to work on when it comes to the issue of progressing formulation of our antimicrobial agents. But I still have one use I want to put on. It's a use on in the progress of my research. So, but we can't discuss that now. The work is still on it. Though I have some results now that I'm working on, but I have some reasons of not presenting that now. But specifically, I've given you, verbally what's our observation, which means that when we use it separately, it has some kind of a little antimicrobial activity against the multi-drug resistant isolate. But when you use it in synergy with plants, there's an improvement on its antimicrobial activity. Then now, the third thing, the one we did recently, which has to do with the use of nanoparticles, it gives a kind of a very fruitful result, in eliminating of most of these multi-drug resistant strains. So I think that has to do with the specificity that you may need to know as regards the research we've done so far on that. Thank you.
Thank you very much, sir.
Yeah.
Thank you so much.
Yeah, thank you, sir. And there was a question from Nahida Fatima. She asked, sir, can you give me the examples of sources apart from crustaceans for isolation of chitosan? So that was the question from Nahida Fatima.
Suita ma'am,
Yes, sir.
now after answer of this question, you conclude, please.
Yes, sir.
The question is in the chat box, Dr. Ajay.
Yeah, it's in chat box.
Okay, I think we are still awaiting a question.
The question is in the chat box, please.
Okay. In the box. Any plant sources?
Correct. For chitosan.
Okay.
For chitosan.
Isolation.
Okay. So can you give me this example of sources apart from crustacea for isolation? Okay. The question is that can you give me the example of sources apart from crustacea for isolation of chitosan? Yes, there are other sources. So the other sources, like I said, is that is something like a snail shell. So if I have to, I hope you are hearing me, please. Hello? So I said the other sources. Hope you are hearing me.
Yes,
Okay.
sir. Yes, sir.
So the other sources include snail shell, which I said I work on. Hello?
तुला माझी काही मदत नकोय नक्की आणि कथा तिथं बसून मदत करते तुला आहे की नाही येस आता पुन्हा अर्ध्या तासाने पुन्हा एक बिस्किट खायचं अर्ध्या तासाने की एनर्जी
Hope I can continue with my illustration and explanation. So based on the question, okay, yeah, thank you. So there are other sources, which I said that apart from crustacea and plant, I said that the other sources we include snail shell. The point is that, well, physically, I even have some because I'm currently in my lab for the production. For example, now the first experiment I did, I extract from the plant and snail shell. But the industrially manufactured one that I work on last is from shrimp shell. If I can even, you can see it here. I just try to make it physical. So because this one now we prepare it takes us about 2 days to prepare. That is from industrial source. This is the one that I did for the last antimicrobial test, and this one too was very effective. And it's the one that I coupled with the nanoparticles, which I find to be very much effective. Then the last one, because based on your question, you are talking of some other sources. If you may not be able to see clearly what I write here. This is a snail shell. Snail shell. So this one now, I extract it from snail shell, and it was very much effective too when we try it. But the issue of the antimicrobial activity per se, of the Chitosan is very low, because we don't count it as a drug. And that's why I said that it is very good to use it in synergy with some other plant sources or other bioactive sources. And well, it has so many antigenic advantages, because even now in the plaster we use for wound, we have Chitosan in some of them, whereby it will prevent anti-inflammatory condition, and it will be able to dry up the wound fast so that the wound will heal. So in some of these plasters, some of the components are Chitosan. So if to answer your question directly, I said that prawn and shrimp, prawn and crayfish are crustacea. Then shrimp too is part of crustacea. But apart from crustacea that you said, we can also get it from snail shell. Though snail shell, the shell contains more of calcium carbonate. But at the same time, based on previous investigation, people have extracted it from there, though there is positive information on that, and I've tried it too. That's why I said that this is the one I did from my lab. So if you can see it clearly from there, this is from the snail shell, and this one is not really jelly-like that of the shrimp shell. And to get this one too, it's a very stubborn, because the snail shell are very, very hard, and it's not really easily degradable like the shrimp shell. I think you can see the one from the shrimp shell is jelly-like. Even though there is possibility of adding another chemical to make this one to be in a bead form. So I think it's a kind of a progress in the scientific realm when we can be able to get some from some of these sources. Though I also learned that the bird feathers too can also be a source. So in essence, those are some of the typical sources that you can get synthesized Chitosan apart from the crustacea. Thank you. So that's why I showed the sample from my lab. This one is from snail shell, and we tried the activity too. Thank you.
Thank you, sir. That was really very elaborated explanation we have. Thank you very much, sir, once again from everyone. And on behalf of Microbiologist Society India, we thanks a lot for sharing your valuable research glimpse and your valuable time also. So with this, we conclude the session, and I thank everyone to participate in the session. And special thanks for Professor Evo Ajayi for giving his valuable lecture. Yeah, thank you very much, sir. Have a good day.
Okay, thank you. I appreciate it.
Thank you. Thank you.
Thank you. God bless.
Thank you.
Yes.